Hello, and thank you for joining us for Chardan's Virtual Ophthalmology Summit, where the focus of the current installment is on rare ophthalmic diseases. My name is Daniil Gataulin. I'm one of the senior biotech analysts here at Chardan, and it is my pleasure to introduce Zandy Forbes, CEO of MeiraGTx, to our next session. Zandy, thank you for joining us today.
Thank you so much for inviting me.
Yes, of course. So the format that we'll follow for the session is the fireside chat. With that, let's go ahead and get started. So first, if you could spend a few minutes and introduce yourself and Meira and its key programs to the audience.
I'm Zandy Forbes, the CEO of Meira. Meira is a genetic medicines company established about 10 years ago. We're a public company since 2018, and we've focused on using genes in multiple ways, not only gene replacement, to address some of the unmet needs that exist in other therapeutics. So we have focused on areas where we can, with very small, locally delivered doses, have a disease-modifying effect or a big physiological change on the target tissue. We have 4 late-stage programs, 2 in the eye, 1 in radiation-induced xerostomia, which is in a pivotal phase 3, it's a pivotal phase 2, and 1 in Parkinson's using glutamate GABA change to change the circuitry of the Parkinson's brain, also about to go into a phase 3. And very importantly, which has allowed us to be in these late-stage programs, over the last 10 years, we have built end-to-end commercial-ready manufacturing. And what that means is we have a platform process which has been vetted by the FDA many times, and that has been made for multiple viral vectors. So I think 20 or more viral vectors. We have a plasmid facility under GMP. We have 2 GMP viral vector facilities, all single-use philosophy, so you can change the size of bioreactors overnight. Every room has its own air handling, so it's as if it's its own facility. And in order to be able to launch a commercial product, we actually built a QC facility which has a commercial license, not just a license for clinical, that allows us to test and release the materials that we make. All of that has supported our late-stage programs and allow us to be either BLA ready or phase 3 ready. And it means when we file an IND with our own material, that material is essentially similar to what will be commercial. So we, in addition to that, we have developed a way of controlling genes, any transgene, the expression is controlled by a small molecule, which is oral. We have multiple small molecules, and we've done this for antibodies, peptides, hormones, GLP-1, leptin, a whole load of different biologic therapeutics, which we can now make the body make when you take a pill, rather than make it outside the body and inject it.
Got it. Excellent. Thank you. That was a great overview. Certainly quite a broad pipeline, and I'm sure we could spend hours talking about your different programs. Of course, the focus for this summit is on ophthalmology, so that let's focus our discussion here. Recently, over the past several months, you've made a huge regulatory progress on your LCA4 program that could potentially make it first to market among Meira's wholly owned programs. So before we get into the construct and the data for this program, can you explain to us what LCA is, leber-congenital amaurosis, what is the subtype for, and potential addressable population here in the US as well as Europe and the UK?
So just very simply, yes, LCA overall is a category of inherited retinal diseases. LCA covers, for example, RPE65, for which Luxturna was approved, and it tends to be quite aggressive, appearing in young kids, and the individual genetic forms tend to be pretty rare. The form LCA4, which we are discussing now, is arguably the most severe and the most aggressive of the LCAs. And it is the result of the absence of function of a gene called AIPL1. Just so you understand the rarity, there's maybe a few fewer AIPL1 patients than RPE65, maybe half. It's not really known because more patients come out of the woodwork, obviously, when you get a therapy. But very aggressive. Children are born blind. That means they have no ability to see an object moving. It's called no hand movement. And it's very obvious that they're blind. LCA4, AIPL1 kids exhibit what's autistic-like behavior because they've never seen, and they're essentially born blind. Within 4 years, the entire retina degenerates. So it's not only extremely severe, but should there be a treatment, it has to be done within the first 4 years of life. Now, it is ultra rare and very severe. So we at Meira have in our manufacturing facility, we have something called a specials license. And that allows us to release material to the UK for physicians to use to prescribe outside of a clinical study. So this could be something that a physician has seen preclinical mouse data and really thinks this could help a severely affected child or individual. And we decided, in collaboration with a charity at the Moorfields Eye Hospital, to release AIPL1 gene therapy to replace the AIPL1 gene to hospitals in the UK to treat the kids. So this was initially done at Great Ormond Street Hospital, where 4 children were treated in one eye, and all under the age of 4, and one eye because they wanted to check the safety. And these were indeed children. So while they were blind, you nevertheless need to be cautious. So those 4 kids were treated, and every single one of those children could see with the treated eye, and the other eye degenerated as those children got older. And this was completely remarkable. So this means that they weren't just vaguely seeing. These very young children who'd had the AIPL1 gene replaced can now see, they can color, they can navigate around the world. Their behavior changes to that of a seeing child. And this was such remarkable data that we had sufficient material to release enough material. We found every vial that we could release outside of those you have to test for quality, and it allowed 7 more children to be treated, but this time in both eyes. That meant that 11 total children were treated, 100%, all 11 saw what I described for the first 4 in the single eye, all went from blind to being able to see with those changes in behavior that were described in the first 4 children.
Yeah, it's quite fascinating. Those results are quite fascinating, and I'm sure it means a world to both those patients and their caretakers. The change that they've gone through. A couple more questions about the disease and its diagnosis. Is it something that is identified as part of the newborn screening programs?
So newborn screening programs only contain genes for which there's a treatment at the moment. So we have been in discussion with various newborn screening programs, and this will go on, certainly in the UK now, and hopefully in the US, when it is approved. So yes, this is absolutely something that should be on every newborn screening panel. It's currently, the test is valid. It's on the inherited retinal disease panel that if you are blind, you get tested with. So given that I said these babies were born blind, they are found because it becomes evident quite rapidly if you have a child whose eyes are moving strangely and who is unable to see. So they then get the IRD panel and they're identified as having AIPL1. So it does tend to happen early.
Got it. Okay. So let's go back and talk about the data. So as you mentioned, first 4 patients were treated unilaterally. Then consequently, 7 more were treated in both eyes. The data from the first 4 patients, I believe, were published in Lancet earlier this year. Can you walk us through key results? What specifically? I mean, I understand, going from no visual acuity to visual acuity is a huge step, of course, but more granular. What specifically did you look at, and in terms of both efficacy as well as were there any safety signals?
Okay, so this was really as all first studies are, this was it wasn't a sponsor. We were not sponsoring this study. Okay. It did not have a commercial sponsor. We released material under our specials license. The regulations around specials mean that individual physicians need to write a prescription for an individual patient. So it was not structured as a clinical study. It obviously had all the features of a study, such as consent, such as data collection, etc. Want to address your question of what was observed. Remember, these children were all treated under the age of 4. And some of them, like when they could not yet speak. So the normal endpoints that you would use in ophthalmology, like retinal sensitivity, right, could not be used across all patients. So endpoints such as acuity measured for kids was measured, and I can't remember the logMAR changes, but essentially the physicians at the Moorfields, at Great Ormond Street, at Evelina, they use standardized endpoint measures that are adapted to children. That doesn't mean every child was measured exactly the same. One of the very important aspects of the data is the videos of the children completing tasks before and after treatment. For example, and I believe this is, it's on our website, it was released by the Moorfields, when the data was released. There is a young girl called Arden, and she's in the video. And I think she was treated maybe as a 3-year-old. She was one of the first 4. And you see Arden a few years later, a couple of years after treatment. Remember, she's only been treated in one eye. So the other eye she's completely blind, is degenerated. And so she has a pair of glasses on where the good eye is covered. And the task is one of these validated tasks for children to pick up a grain of sand that is either 3 millimeters, 2 millimeters, or 1 millimeter that the physician drops on this big table in front of her. So when she is using the untreated eye, you can see her. She's trying to lift up her glasses and sneak a peek with the eye that she can see. She can't find it. She doesn't want to be dealing with this. Then she wears the glasses where only her treated eye is exposed. The other one has tape over it. And she's kind of casually sitting there, and the physician puts a 1 millimeter grain of sand, and we've got the videos of all of the measurements, but a 1 millimeter grain of sand. And Arden kind of sits there casually, vaguely looking around, goes like this, puts her finger on the 1 millimeter grain of sand directly, picks it up like that, and drops it into her physician's hand. Like, you know, don't ask me to do something so ridiculously easy. I couldn't see the 1 millimeter grain of sand. And then you have videos of her navigating through the hospital, finding a chair. So it's those sorts of things that are used by ophthalmology physicians to show a change in acuity, right? So they changed by multiple lines on a chart, essentially, the same number of logmar, but also behavior, navigation, reading, recognition of objects. And all of those are recorded in videos, which have been to the regulatory agency. And there's special formats that you have to put them in and all those sorts of things. But those are critical.
Got it. Yeah, of course, that was the concern when you're dealing with such young children who sometimes may not even understand the tasks that they're given, let alone be able to perform it. So, yeah,
There
appreciate
are
it.
courts at the Moorfield who are expert in dealing with children, particularly for looking at changes in the cortex. I mean, they're really, really good over there. So they use state-of-the-art stuff for analyzing kids with ophthalmology problems.
Yeah, got it. Okay. In terms of efficacy, so this is a rapidly degenerating disease, right? By 4 years on average, most kids have lost the ability to rescue any vision, right? The retina completely degenerates. When you're looking at the at those children from, I don't know, several months old or 1 year old to 3, I don't know, I can't remember how old the oldest patient was who was treated. Do you see clear correlation between the age at which the therapy is administered and the magnitude of effect?
Interesting question. I don't know. I don't think so, because there is some heterogeneity between kids, even of the same age. It's not just totally identical for every kid. Only 11 were treated from all over the world. And I do not believe there was a correlation between response and age within that very narrow window. So you probably wouldn't treat a child under 6 months of age, right? In this physician-led study, I'm not sure that people were treated under the age of 1. So you're really looking at a 1.5 to 2 year window in this study.
Okay, makes sense.
But overall, the question remains, this is the largest efficacy ever seen in any gene therapy, right? Or any ophthalmology intervention, basically, with this sort of disease. And it was startling to the physicians at the Moorfields who've seen all of these diseases and done many studies in many indications, the incredible transformation that occurred. And so clearly it suggests that if you are forced to treat really, really young, let's just say 1 to 3 years old, you are at a moment where rescue can be really strong. Normally, like RPE65, when something's degenerative, rather than being born blind, you wait till the children start to be a little bit blind, right? And then treat them. And that can be 11, 12, 13, 14, 15 years old. So because you're forced to treat at probably the earliest stage possible, that may underlie the incredible degree of efficacy, right?
And among those first 4 patients, are all of them past their 4 years where the contralateral treatment?
Oh, yes, none of them got a second eye treatment.
Yeah. Okay, got it. And all of the other 7 have, correct?
Yes. So they all received their second eye within the standard couple of weeks of the first eye, as in the RPGR study, RP65, the standard steroid regimen for kids. So yeah, they were all treated at the same time, but obviously a number of weeks apart, or, you know, as soon as they were able to schedule coming for their next treatment. Yes, every one of them.
Got it. Okay. And appreciating that this is not a standard clinical trial with scheduled follow-up intervals and periods. I imagine some of those patients still go back for a regular visit. I don't know if you have data available in terms of durability to show,
Oh, yes.
for patients who, yeah.
For the single eye treatment, I do believe that some of them are 3 to 4 years out, and the effect remains. I mean, there's no loss of sight, right? So we have videos and we have all of that from those single eye treated. The bilateral treated have not yet reached 3 or 4 years out, but there's no suggestion in any of the kids that once they can see that even 3 or 4 years later, there's any lack of vision. So we do think that we've treated, they're not degenerating like they were before. Clearly, in the case of Arden, she can see really well with the treated eye. The untreated eye, it's gone. So those unilateral treated kids suggest persistent ongoing efficacy, which is exactly what you'd expect, versus the contralateral eye. Obviously, there's nothing you can do by the time we reach that point of over 4 years old.
Got it. Okay. So potentially, next level of perhaps a form of optogenetic therapy could become an option later on for those who progress beyond, but that's a different topic, and that's not one of your programs. So, you talked about specialist license, right? That allowed treatment of those children in the UK. Is there a comparable process in Europe, in the rest of Europe, or the US?
Not really, not at the moment, no. This is specific to the UK and manufacturing within the UK and having UK licenses. So we obviously have multiple manufacturing licenses in Ireland, in the UK, and this specials is one of them. It is only for UK physicians to write a prescription. But children came from all over the world. So there were 3 US children that were treated in that study. And I can't remember which cohort they were in. There were children from Turkey. There were children from all over the world. Because remember, this is rare. And they contacted the Moorfields because their kids were blind with an inherited disease. So that's how the cohort of children arose. There's no way of doing that in the US. And indeed, in the US, it's actually quite hard to start studies in children before you've shown safety in older kids. So no, I believe it's unique currently to the UK.
Okay, understand. So another aspect of UK regulatory landscape is you also have an innovation passport designation for APL-1, which comes with its set of benefits, including the big one is the potential to file MAA application under exceptional circumstances without having to do any additional studies. Can you talk briefly about that process and with that designation in hand, where are you in terms of your next steps for this program?
So you are right. There is that pathway in the UK. I think last year we filed this data that I've described of the 11 children and the 100% response rate, as well as the very big effect size with the MHRA. They actually suggested that gave us an ILAP and suggested that we did not embark on additional studies and that we discuss with them filing under exceptional circumstances because of the strength of the data. Another very important aspect of this is, in fact, our manufacturing. So even if an agency accepts that, yes, this treatment in very young kids, let's just say, reverses the blindness and maybe durably in these kids, and that is shown with our data, so you can accept that, it's still absolutely critical from a regulatory perspective that your manufacturing is of absolute commercial quality. And one of the big aspects of the filings, which we're in discussions in the UK, but also with the FDA and the current administration is very positive in this respect, is dealing with a very rare disease and reducing the number of PPQ batches that you need before you can file, before approval. So our manufacturing really allows us to take this, we'll call it exceptional data, and that those are the exceptional circumstances, and move forward with a manufacturing plan and indeed material that will be used commercially in a way that is curtailed and slightly altered compared to a large indication or a less degenerative indication. So there are two aspects to the discussions on regulatory. And in both jurisdictions, we're having very similar discussions and positive discussions about that aspect, the CMC aspect, which we wouldn't really be in the position to do unless we were the manufacturer, unless we did the assays and validated them and had the process in-house.
Okay, I understand. Yeah, that makes sense. There's been some recent positive messaging from the FDA, including the roundtable discussions focusing on potential accelerating access to drugs for high unmet need for which there are no treatment options. With that being said, and with the nature of LCA4, would you say these discussions are starting to provide you clarity with what the pivotal program is going to look like and what the next or clear path for the next steps in the US?
There will not be another study. You said pivotal program?
No, pivotal process. Like what are the next steps to get this on the market?
Well, one of the reasons for specials is because this is so early, so rapid, and so rare. It's not actually feasible to do a sponsored study, to do a clinical study. So that would delay an approval by many, many years, and it wouldn't actually provide more safety. So we're not looking for, in fact, a new study is not really feasible. So we are in discussions, iterative discussions with the MHRA and also the FDA, and they've been very similar in their view of the data and how we need to present it, right? We know that, and the strength of the data has been recognized by both agencies, and the approach to CMC for these sort of rare diseases has also been similarly discussed on both sides of the Atlantic. Now, with respect to the agency, yes, I've been in the meeting in DC with the leadership of the FDA, Dr. Makari and Dr. Prasad and others, but those in particular, the day of the gene therapy and cell therapy public meeting. And in that public meeting, in fact, the new head of the NIH mentioned as an example of the types of drugs that they were keen to approve, a drug that made blind babies see. Now, he didn't say it was AIPL1, but to our knowledge, this is the only treatment ever to make blind babies see. So just in that meeting, which is public, there was a recognition that they are keen to be able to be in a position to have filings for drugs that treat very severe diseases, right? Including diseases of the eye.
Okay.
I would say that discussions with Dr. Makari and Dr. Prasad separately, and I've heard this from many people, have been very positive in the area of gene and cell therapy, particularly in the manufacturing arena, particularly in the way they want to approach endpoints, what trials are required, and using, as we all hear, good science and good sense. So from my perspective, the leadership have been extremely positive about gene and cell therapy for rare, inherited, and severe conditions. And I think I'm not the only one. I think that's generally accepted, that they are putting a priority on those sorts of treatments.
Okay, I understand. And given how quickly this program is moving through the regulatory discussions, have you started internally thinking about the commercial strategy? Have you initiated the pricing and reimbursement negotiations or discussions or potential partnerships to bring this to market?
Yes, we are thinking about all of that. And we, as I've mentioned, we obviously are always in discussion with potential partners, and that remains true today. So that's absolutely having a great drug that works really well, that you've manufactured and is sitting there, is of absolutely no use unless you are able to give all the children born with AIPL1 in the world access to that drug. So with that in mind, we are in many discussions to make that happen.
Got it. Okay. And maybe in the next couple of minutes, I know we have a couple of other programs in inheritable retinal diseases. Some are more advanced, some are less. But among some that are listed on your pipeline are RP65, achromatopsia, CNGB3, and CNGA3, and some of the other early stage programs. Can you give us 30-second, 1-minute overview of where you are with those programs?
So we currently focusing on our 4 late-stage programs. So our pivotal program for xerostomia, our phase 3 for Parkinson's, and the BLA-ready programs and manufacturing for commercial. So we aren't currently moving forward our other IRD programs. RPE65 had very good data. It is phase 3 ready. A3 and B3, similar, they are ready to go into a phase 2. All of this we can manufacture. We have preclinical, arguably the most potent Stargardt's program using superior inteins. So that is ready for an IND. And we also have RDH12 ready for an IND with material manufactured at Meira. So those are ready, but we're not focusing our spend on those right now because we are really focusing on the large indications in late stage development. One program is actually going into the clinic right now, I believe this month, maybe this week, and it is another specials. So BBS10 is a specials we've been working on. We designed the vector in collaboration with the charity, and there is a physician currently in the UK who has BBS10 kids and is writing prescription for BBS10 to potentially treat them in the very near future. But we aren't embarking on the other programs until we have a partner.
Got it. Okay. Yeah, makes sense. I also wanted to spend a few minutes to talk about the XLRP program that you sold the rights to J&J back in 23. So we know that J&J have reported data from the phase 3 trial earlier this year in May. Um, the trial did not meet the primary endpoint of MAZE, but it looked quite positive on the other endpoints. And I wanted to hear your thoughts on the discussions of the importance of MAZE for XLRP and from the regulatory standpoint, what kind of hurdles do you think J&J would have to go through to bring this program forward?
So I obviously don't know. We are no longer party to regulatory discussions with J&J, with the regulators. We saw the data at the same time as everyone else at the FFB meeting. And while the primary was not met, the MAZE endpoint, we can discuss how that came about. You are right. All of the other endpoints were, I will say, better than expected and extremely consistent. And overall, 40% of patients responded, like had 2 or more positive domains and 0 in the untreated. Most striking, and we had physicians texting us about this, was that acuity in low light improved remarkably, not just a little, but 2 to 3 lines in many patients. That's 2 to 3 lines on a chart, which is approvable for things like glaucoma in Europe. So we have also previous discussions with the European agency as well as the FDA. We know that the MAZE endpoint was designed and made primary in alignment or to align with the desire of the FDA at that time. The way that the statistics and this notion of 2 light levels was put into the MAZE was designed with the FDA. And we also know that the MAZE was not necessarily critically important to the Europeans, and retinal sensitivity and visual acuity are. So you've also, I'm imagining, have seen the letter from the Foundation for Fighting Blindness that was put out about this data, that was signed by 30 or 40 of the KOLs, many of whom had been in the study, as well as providing patient feedback on the benefits that they've seen. So we feel that the data that has been presented to date is something that would be viewed positively by the Europeans. In the US, there was 3 studies. Remember, a phase 1 where the control was the contralateral eye. The phase 2 where there was randomization of patients that were treated in one eye and then the other eye, the untreated patients, both eyes randomized. And then the phase 3, which was blinded, 2 doses versus untreated. So there are 3 studies supportive of approval. We think that the Europeans are most likely to look upon that very positively. We don't know about the FDA's discussions, given that the primary missed, but nevertheless, all of those secondaries look extremely positive. I will just say one more thing about this, is that this is a global disease, and the study, the phase 3 study, was enrolled in the US and multiple sites across Europe. And if you look at the distribution of patients globally, there's probably twice as many or a lot more in Europe than there are in the US identified. So that's all I can really say, because I'm not privy to the actual discussions, but I believe J&J has said and continue to say to investigators, to the FFB, and maybe to patients who call them, they're in discussions with global regulators.
Got it. And I think it's important to note as well that a competitor's phase 3 program does not utilize MAZE as a primary endpoint.
Yeah. When we and Johnson & Johnson were dealing with the FDA, the people at the FDA who rather demanded the MAZE and demanded the way it was to be measured may not be there any longer. So I don't know the negotiations with other companies, but I do know that 4 years ago with AGTC, which is pre-Beacon, right, and Nightstar, there were similar discussions to ours. It was a very difficult discussion about retinal sensitivity, which was our first choice of primary, and the MAZE was really what the FDA negotiated. And I have no view into what the FDA has negotiated with other companies and other studies. But I'm imagining that there's different input nowadays.
In the next or in the last couple of minutes, a couple more general questions. One back to manufacturing. You described that really well, and tremendous capabilities that your manufacturing has afforded the company. At the same time, you do have a very broad pipeline, right? As you mentioned in the introductory statements, including some potentially really large indications. Would your manufacturing be able to pivot and support all of those or all of your pipeline's needs?
Yeah, so this is a really important question. And we are asked constantly about what scale is your manufacturing? That's a slightly old school question, right? Because biologics you make in fixed bioreactors, a giant like fill a whole room. For our therapies, we followed regulatory agency guidance on the architectural design, the floor plans, and the people flow, and the use of our facilities. Our facilities were built based on the guidance that manufacturing facilities for gene and cell therapies of the future should be single use. What that means is there are no fixed bioreactor sizes. So our suites are built to hold 50 liter, 40 liter, 80 liter, 2,000 liter. So you can run an entire process, 80 liters, right? Make enough for an entire clinical trial, wheel out those 80 liters, throw them away, and wheel in 2 days later, 500 liters, 2,000. So we are scalable in time. Each room has its own air handling. So you switch it off and totally clean every room like it's an entire facility, which means that it's modular. You can have room A, B, C, D. And guess what? If you need to scale, you put room F, G, H in there, right? Because it's their own air handling. So I hate to say there's no limit to the scale, but we could go to a building next door and put in 10 more suites. We can currently supply at our current scale all of our programs and those programs commercially. Now, there's another important aspect of this. I said that we focus on small doses locally delivered. There are many reasons for that, from an efficacy and safety perspective, to address the local cell type by injecting into that site seems to have resulted in very good efficacy, ultimately very good safety, but low dose of a highly potent product means you have to manufacture way less. Our costs of goods are really, really low. So in the single digit thousands for Parkinson's, for example, we're talking about large indications because the dose used in Parkinson's is tiny, like 35 microliters of a 1E12, for example. Very, very different from a total of 5E15. So that impacts cost of goods, manufacturability, time to manufacture, and all of this using a process that can accommodate, well, it was built on 20 or more viral vectors that can accommodate, we'll say, for the moment, any viral vector and get it into commercial like GMP in our facilities in months. So there's more to manufacturing these sorts of products than how big are your bioreactors. It's about flexibility and scalability in time as well as in space, right? I mean, by space, I mean, can you add more units? And so we can make enough material for all of our programs and in fact for other people's programs. And for example, we are commercial manufacturer for Johnson & Johnson for the RPGR program. And it's very important, this notion, because you don't want to have a facility that with massive great bioreactors that you can't make a clinical batch in. And yet you also don't want maybe to have a completely different process for every different virus, because then it's unmanageable. So platform process, make our own plasmid, single-use philosophy, scalability of that platform process, and really importantly, for releasing commercially and into the clinic, we do our own QC, validated QC using our process development group that we do in-house with a commercial license. So we can release things in 8 weeks, whereas the vendors out there that we do use and have been using, the likes of, I don't know, Wuxi, BioAlliance, Charles River, those sorts of vendors, find it very difficult with their workload to have that sort of quality and time frame. So all of that put together allows us to be end-to-end and scalable and have every assay as well as the process in our hands, so that when we write the FDA documents, the CMC section, as you know, is the biggest, right? We have written many of them, and every aspect of that, it's our responsibility to write. And our quality systems oversee the whole company, but we have very, very strong QA leadership. Our leader came from Novo Nordisk, and the audits we have across the board in CMC, in clinical, sometimes have 0 findings, sometimes 1 minor. So it's the entire infrastructure that allows us to supply clinical and potentially commercial at the scale that is needed at each of those stages. Much harder, you have to go to different places for those different things.
Okay, yeah, got it. One last question that we like to end those discussions with. But of all the things that we talked and maybe some that we didn't, what do you think investors are still missing about MeiraGTx's story?
This has been an interesting 4 years for biotech, and to the largest extent, the behavior of stocks has not necessarily reflected the events, the successes of stocks, right? Interest rates, economy, FDA, there's been an overarching pressure, downwards pressure on stocks. We, from our investors, we have investors that are extremely excited about xerostomia, which we've got FDA feedback They aligned that our current phase 2 multi-arm study, double-blind, is pivotal in their eyes from a CMC perspective, but now also from a clinical perspective after our RMAT meeting. We have a lot of interest there. It's a large market, and it's almost like a pipeline in a product, right? Radiation-induced xerostomia, but then there's the radioligand-induced xerostomia. Is there a potential use there? There's lupus. Is there a potential use there? So we're getting increasing interest in that. And we obviously have interest in our riboswitch program, which we're in discussions initially with the FDA about taking that entire platform into the clinic. Our first example might be one of the metabolic peptides that could target rare disease as well as very large indications. So that's another area of interest, this unprecedented technology to control the delivery of any biologic therapeutic from a gene that's a one-time delivery with a pill and precisely dosing it to exactly the dose you want every day. So there's lots of different interests, obviously AIPL1, obviously partnering, but I can't say that any one of those things drives the value of the company, the stock price of the company. And I would suggest that all of them combined result in a valuation that is multiples of our current, even though our stock has performed quite well in the last month. So, it's such a hard question. What do investors see? Because investors themselves have a lot of other considerations, right? Their performance, their redemptions, what's working, what isn't working, their portfolios, and liquidity. These are all issues that investors and companies deal with. And it's been a very long 4 years, 3.75 years that this has been happening. So we're all, working to see, I actually see a little bit of a change in investors' mood and attitude recently. I think people are looking for these undervalued diamonds that are out there in the kind of wasteland that's being created. And I think investors are really doing a lot of work on what do they want to own, not necessarily today, but over the next few months and as we get into the end of this year. That's my sort of gut feeling. And, I could be 100% wrong. And the world is, a bit volatile, isn't it? So hard to say what investors are missing or not missing.
Yeah, but no, I appreciate that answer.
It's just
Okay. All right. I think with that, we are coming to the end of the session. Again, Zandy, really appreciate you taking the time to join us for the summit, and look forward to hearing more updates on your other programs.
Great. Thank you so much for inviting me.